Hello dear student, today we are going to discuss type of cell lines, virus cultivation, primary cell line, secondary cell line, continuous cell line, individual host cell line. After watching this video, you will be able to discuss different type of cell lines. So first of all, we know that viruses are obligate intracellular parasite. It means that it cannot be cultured on artificial media. Viruses need living and metabolically active cells, host cells. Their cultivation is possible due to animal culture. It means that we should perform animal culture. We will provide living cell to the virus so we can then study the virus and we can prepare various type of vaccines to we can diagnose the viruses, and this is only possible due to the animal culture. Now, what is actually cell culture? Cell culture mean that suppose we take the cell from animal and we grow this cell in artificial media. Now this is known as animal culture. We in vitro culture of animal cells or animal tissue or animal organ is known as animal cell culture. For the animal cell culture, first what we can do, we mincing and treating the animal cell with enzyme. We take the cell and we treat it with enzyme. Due to this enzyme, it's separating the tissue to individual cells. Now the cells are suspended in the media, and we can also add antibiotic to forward purpose to prevent the growth of bacteria. When you provide all these material and you separate the cells, this cell will form a uniform layer uniform layer on the surface. Now this monolayer, this is now called primary culture. What does primary culture? When we take the cells from the tissue and culture of the media, this first cultured grown cells is called primary cell culture, and it can be subculture. You can take it and you can subculture it by providing fresh media. This is known as secondary culture. Now what is cell line? When tissue are completely digested and the individual cell are mixed with viral growth media and dispensed in the tissue culture plus, this is known as cell line. Suppose we take the tissue, and this tissue is grow on the media, and when this tissue is grow on the media and you collect the cell, this is called primary culture, and this cell is treated with viral media and suspended in a flask, then this is known as cell line. The primary cell line is actually freshly isolated. It means we take the primary cell line from the primary cell culture. Now what are the property of primary cell line? They undergo limited division 5 to 10, possess diploid chromosome, मतलब mean 2n chromosome. And example of the primary cell line are rhesus monkey cell line used for myxoviruses, enteroviruses, and adenoviruses, human amniotic cell line, and chick embryo fibroblast. These are primary cell lines, but the main problem of primary cell line is that you should subculture it. You cannot maintain its generation. it is limited growth division for 5 to 10, and it you should maintain it in the lab. So to maintain the cell line, you should perform secondary cell line. Secondary cell line are actually obtained from subculturing a primary cell line. You should take the primary cell line, and this is subculture and the media, and this is called secondary cell line. Secondary cell line undergo moderate cell division as compared to primary cell line up to 5 to 10 divisions. They possess diploid chromosome. It includes human fibroblast cell line, and which is used for cytomegalovirus, MRC5 and WI38. Actually, these are human embryonic lung cell strain. You know that what is cell strain? When you positively collect cell line, it is called cell strain. Used for the preparation of various viral vaccine, for example, rabies, chickenpox, hepatitis A, MMR. MMR mean that mumps, measles, rubella vaccines. They also support the growth of human simplex virus, varicella zoster virus, cytomegalovirus, adenoviruses, and picornaviruses. Now, continuous cell line. What are continuous cell line? They are derived from cancerous cells, hence have independent division and possess haploid chromosomes. They are easy to maintain in the laboratory by serial subculturing, hence they are widely used as compared to primary and secondary cell line. Example are HeLa cell line. HeLa cell line are human carcinoma of cervix cell lines. Hep2 cell line, these are human epithelioma of larynx cell lines. They are used for RSV, respiratory syncytial virus, adenovirus, and herpes simplex virus. KB cell lines, actually human carcinoma of nasopharynx cell lines. Mucous cell lines, they are actually derived from human synovial carcinoma cell lines. It is useful for the isolation of viruses as well as chlamydia. Chlamydia is intracellular parasite, it is bacteria. Virus cell line. Virus cell line mean vervet monkey kidney cell line. Vervet monkey kidney cell line and used for the production of rabies vaccine. BHK cell line. BHK mean baby hamster kidney cell line. Detroit 6 cell line, actually it means sternal marrow cell line. Chain CILK cell line, actually human conjunctiva C, intestine, I liver, L kidney or K cell line.